Hello, everyone. My name is Victor Manley, and I am a graduate student in the Halmos College of Arts and Sciences, Health Studies concentration. Today, I'm going to talk about microscopy. Let's talk about the two main types of microscopes. Your first type of microscope is your light microscope, which uses the visible light spectrum in order to visualize specimens. This is the main type of microscope that we will be using throughout our undergraduate studies. There's two main types of light microscopes, in which the first one is a compound microscope, as seen here. It has a magnification of up to 1,000 times and is best used for small objects such as bacteria or tissue samples. This is the main type of microscope that we will use in Biology 1 and in the upper level science courses. Both the compound and the dissecting microscope here are able to have both an ocular and an objective lens. But the dissecting lens can magnify up to 300 times, but it's better used for larger samples such as rocks and insects. And you will see that in biology too. The other type of microscope is the electron microscope. And that uses a shorter wavelength with electrons in order to visualize specimens that are dead, but can magnify up to at least 1,000 times and much greater magnification than light microscopes. The two types of electron microscopes are the scanning electron microscope, which gives a 3D looking image of the surface of the sample topography, such as this, and your transmission electron microscope, which gives more finer detail on the internal structure of cellular components. Talk about the anatomy of the microscope. First, you have your eyepiece, which is used as your observation or ocular lens, used to observe specimens and enlarge images that are formed on the objective lens. You have the lens tube that holds the lens that is used to magnify the specimen. The objective lens holds and focuses and enlarges the specimen that's on the stage. You have two adjustment knobs in which you have your coarse focus, which is your large knob, and your fine focus, which is your smaller knob. The stage supports the slide, and the clips on the stage hold the slide in place. You also have your diaphragm, which allows light to come through and shine onto the objective lens. The mirror, which reflects light onto the objective lens. The condenser here focuses light onto the specimen. The base, which supports and stabilizes the entire microscope, and the limb, or the neck, which is used to hold and carry the microscope, in which you will hold the base with one hand and hold the neck in the other while transporting your microscopes. So let's talk about some main properties of microscopes, in which the main property is magnification. That is the size ratio of the observed image to the actual image. An example of this, when you have a 50x magnification, that means that your observed image is 50 times larger than the actual image. The other property of microscopes is their resolution, which is the ability to distinguish between 2 points clearly, as you can see in this picture here. And resolution is determined by the magnification, the lens quality, the viewer's visual acuity, and the lens's diameter. The third property is the focal plane. You'll need to use, when using at higher magnifications, you will see increasingly thinner sections of light, in which you will need to adjust the stage accordingly to bring the image into focus at those higher magnifications. The fourth property is contrast, in which you need to visualize a transparent specimen using a staining such as methylene blue or iodine. And the final thing I want to talk about today is wet mounting, which is used for viewing the live specimen. So you'll drop a drop of deionized water onto the slide, place the slide on the sample onto the drop, and use the stain in order to visualize that sample. I want to thank you all for your time today, and you have a good day.